I am Thusitha Rupasinghe from Metabolomics Australia, the University of Melbourne. My presentation is mainly focused on investigation of the salinity tolerance mechanism in barley roots using lipidomics platform. Salinity in Australia. Due to the increase of world population and global warming, our food production is facing many challenges to fulfill the high demand of food. In Australia, approximately 53% of total land area is used for agriculture. Salinity and droughts are the major threats to agriculture industry and the crop yield. In Australia, our farmlands has been affected heavily by the drought and the salinity. As an example, this is a picture of a valley in Australia, in Western Australia near Bruce, and it is clearly showing the effect of salinity in farmlands. How salinity is affecting to our agricultural productivity in Australia. This is a map of Australia, and yellow highlighted area is the potential land area, and also you can see the red color highlighted area is the salinity affected area, which is salinity level is from 1% to 10%. The upper right hand corner is for the map of the wheat belt in Australia. Most of our wheat and barley farms are located in the wheat belt. Unfortunately, salinity is affecting the same area. The lower right hand corner shows the effect of salinity in farms and reduce the plant growth, which will result in reduce of grain yield. Why we chosen barley for this study? Barley is the second highest grain produced in Australia and approximately 8 million tons per year, occupying 4 million hectares. Australia has the highest quality barley. It's main and also mainly used for malting, brewing, distilling, and feed. We make up to 30% of the world malting barley trade and 20% of the world feed barley trade. Due to the increase of salinity and drought, our wheat and barley export is in danger, approximately costing us $6 billion per year. How salinity affect on plants? Osmotic stress will reduce the water around the roots, and that will result the reduction of root turgor pressure, which affect the root elongation and root cell division. This will trigger the signaling to shoot and reduce the shoot growth. However, some plants can tolerate the osmotic stress and shown resistance to salinity and grow well in the presence of salt. Here we are investigating the salt tolerance mechanism in plant, and we are using lipidomics platform to find out the defense mechanism in barley roots. For this study, we have chosen two barley cultivars, Clipper and Sahara. Clipper is known to be a tolerant to salt stress, and Sahara is sensitive to salt stress. So this data clearly supports that how the salt stress affects on root elongation. Clipper shown only 36% loss of root growth in the presence of salt compared to 50% of Sahara when we introduce the salt stress of 150 millimolar sodium chloride. So we have seen a clear difference between Clipper and Sahara shoot growth. Now we want to investigate changes in lipid profile in barley roots due to salinity. Both Clipper and Sahara cultivars grow on agar plates, then harvested in a week time. So we use lipidomics platform. So lipid and fatty acid analysis is carried out using LCMS, liquid chromatography mass spectrometry, and GCMS, gas chromatography mass spectrometry platform. So lipidomics approach. So the tissues are harvested and samples are quenched to stop any further degradation of metabolites and lipids. The tissues are they are homogenized using cryomil, and lipids are extracted using solvent-solvent extraction. Then lipids are analyzed using LCMS, and fatty acids are analyzed using GCMS platforms. To investigate spatial distribution of lipids in the roots, we have used imaging technology. So LCMS and GCMS data is processed and statistical analysis is performed to identify significant lipid species. So the lipid analysis workflow. So we have chosen Clipper and Sara, and we treated with salt with 5 replicates. So total lipid extract using organic solvent and analyzed for lipids LCMS and fatty acid by GCMS. For imaging, portion roots were prepared with suitable matrix to detect lipids and their spatial distribution. So coming to the results, targeted fatty acid analysis using GCMS platform. So this bar chart showing the amount of the fatty acid composition, and C is for Clipper and S is for Sara and Z is for the each zone. So we have chosen 3 zones to work out which area is mostly affected. So the fatty acid profile is significantly different between 2 cultivars and also affected by the salt treatment. In addition, the results showing fatty acid analysis shows in zone 1 has changed due to salt stress. So moving into LCMS targeted lipid analysis, the PCF plot is clearly showing the significant difference between 4 groups, Clipper and Sahara with and without salt treatment. So also in the heat map, 2 cultivars are well separated, primarily based on cultivars, based on Clipper and Sahara, and secondary based on the salt treatment. The top 50 significantly change in lipid species is mainly based on PC and PE class. So to confirm the results from targeted analysis, we have used untargeted lipidomics approach. Here only root 4 Clipper and Sara. This untargeted lipidomics also supporting the targeted lipid data. Yes, the fold change of each lipid species we have identified due to the salt treatment clearly showing some lipid species are significantly changing. The Venn diagram is showing only 89 lipid species are common within 4 groups, and the lipid profile in Clipper showing less effect from salt than Sahara, as only 121 common lipid species in between salt treated and control, while Clipper has 253 common lipid species within treatments. Also, the box plot I'm showing clearly showing the fold change of some lipid species, and we have noticed most of lipid species are changing due to the salinity accompanied with more double bonds. So does lipid oxidation may play in any role of defense mechanism in root against salinity? So to investigate that, in 2016, Seung has published how reactive oxygen species can affect the defense mechanism against salinity in plants. Also, it has been well published oxypin metabolism in response to stress in plants. The question is, does oxidized lipids and oxylipids in barley roots will show any significant changes as response to salt stress? To investigate that, we have employed LCMS platform to investigate the oxidized lipid profile, and oxylipin profile due to soil stress in clipper roots. The reason we have chosen clipper for this study shows the higher soil tolerance capacity and we can get more information of defense mechanism against salinity. So we have collected the data from the LCMS, and so we process the data and the statistical analysis for oxidized lipids, clearly showing significant difference in control and salt treated samples. However, we have noticed that changes in oxidized lipid profile is not simply based on one single lipid class or oxidized lipid class. So the heat map clearly showing a trend, a separation of two groups, salt treated and untreated roots. But most of the oxidized lipid change, and we are currently investigating exact pathway to identify the defense mechanism. So continuing from the previous results, as an example, these box plots are showing the significant difference and similar trend within the oxidized lipid classes. Example, so HOT PC364 and HOT PC365 showing the reduction of the amount of oxidized lipid when treated with salt. However, HPOD keto PC364 and HPOD keto PC366, HPOD keto PE364 increased with salt treatment. Same time, HPOD keto PG365 has shown the opposite trend than oxidized PC and PE species. So this is quite interesting data, the complexity and the mechanism. So it's really interesting results to investigate further to identify the mechanism and the biosynthesis pathway. So also we have analyzed oxylipin. So oxylipin analysis supports the published literature as expected. All the oxylipins are in higher level in soil-treated barley roots than untreated barley roots. This data supporting the hypothesis of oxylipin play an important role in defense mechanism against the soil stress in barley roots. This data is produced by Dr. Cornelia from Professor Ivo Fresner's lab as a collaboration between Melbourne University and Göttingen University. So moving into the spatial distribution. So this is the images of barley roots, treated and untreated. So the control is no salt, and treated with 150 millimolar sodium chloride for 48 hours. The data has been analyzed in positive mode. As we know, the lipids are changing in barley roots when salt stress is introduced. But localizing those lipids is also important to see as images. So I'm showing here PE365 and PC385, which are reduced dramatically, as well as OPDA species after 48 hours of salt treatment. So the second slide also showing spatial distribution and reduction due to the salt stress, mainly with longer chain length with more double bonds as expected, while level of saturated lipid species are higher than treated with salt. So this interesting data is opening up many questions, and we are looking to expand methodology to confirm the defense mechanism in barley roots. This will allow us to modify barley cultivars to increase the salt tolerance. So, as a summary, the lipid profile of barley root shows significant difference between Clipper and Sara cultivars. So, the barley species tolerant to salt stress shows PUFA containing polyunsaturated fatty acid containing PC and PE lipids, and HPOD keto and HPOT keto oxidized lipids are increased, and HOT and HOD oxidized lipids are decreased as response to salt stress. So oxidized lipids and oxylipids play an important role of the defense mechanism against salinity in barley. And pathway mapping of oxidized lipid biosynthesis is important to investigate to understand further insight of the defense mechanism in barley response to salt stress. Finally, I like to acknowledge my supervisor, Professor Uta Rosner, and Dingy Yu and Daniel Sabia, providing most of the samples and data for this study. Um, also, as a collaborators, Professor Ivo Fassler and Pablo and Cornelia from Göttingen University. Thank you.